Assalamualaikum. Welcome again. In the previous lecture, we have started with the enzyme classes and we have discussed various enzyme classes with their uh, functions and some of their reactions as an examples. So we have discussed about the different classes. We have seen the example. These were the material that we have discussed in the last lecture. So now we will be starting about more details of these enzyme classes. Along with this, what are the things that are included in that? What kind of the enzyme that are included in that? So the first class, that was the oxidoreductase. Basically, as the name indicates, that is involved in the catalyzing reactions of the oxidation reduction type of the reaction. It means either it may be involved in adding or removing of the hydrogen atom, electrons, or addition of the oxygen, and so on. And there are so many examples in several kind of the reactions, several kind of the pathways. For example, lactate dehydrogenase, glucose oxidase, peroxidases. It's again a big subclass. Catalyses. It's again a big subclass. Phenylalanine hydroxylase that is involved in the protein type of the reactions. So I will take example of the lactate dehydrogenase. Lactate dehydrogenase basically that is producing the lactic that is producing the pyruvates. Uh, so, so there are so many lactate dehydrogenase, pyruvate dehydrogenase. So lactate dehydrogenase, the substrate is surely that is the will be the lactate. This is the lactate. This is the lactate, and when the lactate dehydrogenase that is acted upon by the lactate dehydrogenase that is converted into the pyruvate. So look at this one. Originally, this is the substrate that is in the reduced form, and this is NAD coenzyme acting as the coenzyme with this one, and this coenzyme that is originally in the oxidized form. So the function of the dehydrogenase is to oxidation and the reduction. So this will be going to oxidized and this will be going to reduced. Originally this is oxidized form, so this will be going to reduced. So after that after that this will be there will be production of the pyruvate, and originally that was in the reduced form, so this is in the oxidized form and this will be in the reduced form. This will be in the reduced form. So the dehydrogenases that belong to the class of the oxidoreductase, so performing the reaction of the oxidation and reduction. Some other classes from the oxidoreductase, this is the main class oxidoreductase. Some other subclasses, these are the subclasses: oxidases, peroxidases, dehydrogenases. This is one of the, you look at the example of the dehydrogenase, so it's the subclass. The second class was the transferase that is associated with the transfer of certain groups, transfer of some of the functional group, methyl group, phosphate group. In the previous lecture we have seen the example for the phosphate group. So there should be some donors, there should be some acceptor. Example is transaminases, transferases, kinases. We have seen the example for this kinases in the previous lecture. Transmethylases, transpeptidases, acyclases. So these are the different examples from the transferase subclasses. So look at this one reaction for the transferase. So one of the subclasses is the transaminase. One of the subclass of the transferase is the transaminases. So this one, first one, this first one, transaminases. So this is specifically this transaminase is the alanine transaminase enzyme that is working on the alanine. So that is working on the alanine, and what is that doing? That is actually transferring this oxygen to this carbon, and this hydrogen and this NH3 transferring to this carbon. So there is a, you can see there is an acceptor, there is a donor. So both of these are the acceptor and the donor. This is accepting from this point, and this is donating this to this one. In this case, this will be the acceptor. So L-alanine and alpha-ketoglutarate that will creating pyruvate, and the L-glutamate, that is an important reaction from the urea cycle, where the amino acids alanine that is converted into the carbon skeleton. So removing the NH3, this NH3 that will further go to the urea cycle. Transaminases that is providing the important functions. Then hydrolases catalyze the hydrolysis of various bonds and add the water across the bond. Hydrolases, as the name indicates, the moisture or the water is involved, and that is used for breakdown reaction. It's a breakdown reaction normally. So you look at this one. This reaction takes place in the presence of the water, and add the water across a bond. So this is important. This reaction takes place in the presence of water and adding a water for the breaking of the bonds. So look at this one. This is the pyrophosphate. So that is converting into the phosphate by breaking type of the reaction, and the water is involved at that point, that is causing the breaking. So there are two phosphates that are developed. So hydrolases again that have several classes, these are subclasses. These are the subclasses: esterases, peptidases, glycosidases. Then the lyases, class number 4, that cause the cleavage various bonds by means of method other than hydrolysis and oxidation. So this is important. Breakdown is here, but this breakdown is due to the water molecule, right? But in case of the lyase, breakdown is there, but that is not due to the water molecule. Water molecule is not involved, or oxidation process that is not involved. Oxidation also causes the breakdown process. So that add water, ammonia, carbon dioxide, different kind of the substance that can be added across double bond, breaking down the double bond so that valency is free. Now these substances that can be added at that double bond are remove these elements to produce the double bonds. So both of the function that is performing these lyases, it can add the double bond or break the double bond. So various examples: fumarase, carbonic anhydrases. So this is look at this example. So breakdown as I told you, that is involved for the breakdown other than water. So look at this one, it's a fructose 1,6 bisphosphate. So it's a 6 carbon compound, and you can see that it's broken down at that point. So one of the member for the lyases is the aldolase. So this reaction that is actually taken from the glycolysis process. Fructose 1,6 bisphosphate that break down into the middle without the water and converting into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So this reaction that is taken from the glycolysis process, and the enzyme is the aldolase. Surely this enzyme that will be belonging to the class of the lyases. Then the isomerases, you are well aware of this terminology, isomerases. So that is creating the isomers. So biochemical activity catalyze the isomerization change within a single molecule. And there are so many types of the isomers. One of the isomers you are well aware, L and D isomers, alpha and beta isomerization. So one kind of the subclass is the mutases. Mutase. This is the enzyme, important enzyme. This is belonging to the class of the isomerase. So that is doing the isomerization function, isomerization reactions. So mutase reaction, the shifts of chemical groups, and the subclasses isomerases, mutases. These are the subclasses for the, all of these are function of the isomers. Look at this one. This is alanine racemase. So look at this one. This is an amino acid L-alanine, L-alanine, and that is creating. This is L-alanine that is creating another isomer of that is the D-alanine. What is the basic function? This L-alanine and the D-alanine that are the mirror images for both each other, for each other. Look at this one, H3 is on this direction and H is on this direction. So after that, H will be in the opposite direction and H3 will be in this direction. So in this case, alanine racemase that belong to class of the isomerases. Alanine is the substrate here, right? Then the final class is the sixth class is the ligase. That is the normally used in the biosynthetic process most of the time. That is used in the biosynthetic processes. So that join two molecules with the covalent bonds that catalyze the reaction in which two chemical groups are joined with use of energy because it's a synthetic process. So there will be use of ATP to synthesize. It's an anabolic type of the reactions. So there are several examples: acetyl CoA carboxylase, glutamine synthetase. Look at this example. This is the L-glutamate. So there will be the ligation process. So there will be the use of the energy in the form of the ATP, ammonium. So glutamine synthetase. This is the glutamate. So glutamine synthetase. That will be producing the L-glutamine by the addition of this ammonia. So look at this one. That is added. That is like a ligation process. So this is the formation of the L-glutamine from the L-glutamate. So energy is utilized. So this ATP that will be converted into ATP. So these are the briefly I have written in a single slide all the six classes: oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. So all the 6 classes I have written here, and some of the enzymes, important enzymes that are belonging to these classes I have written here. You have to remember all of these. And I have also written here the code number that I have told you earlier, different code number. So you must aware of some of the examples of some of the enzymes that belong, these enzymes that belong to which class, and their EC codes along with their standard abbreviation and common abbreviation. So you have to learn this one. You will be have in mind for these examples for different classes. So we will be closing our lectures on the enzyme classes here, and be starting with some other topics in the coming days. Uh, I hope you will be staying at your home during this corona pandemic disease, and you will be caring about yourself as well as your family. So we will be closing our lecture here. Inshallah, we will meet in the next lecture.